Okay, I'm going to go back. Did that start yet? Yep, it did. Okay. Sometimes it takes it a while to start recording. This is the review for transcription and translation, and you should have that. You have either the blank one or you have the one that's completed. Either one you could type on, you could annotate on it, you could jot notes down on a piece of paper, whatever you want, but this would be really good for you to use to study and maybe have it ready when you're taking the quiz. Central dogma. This is this belief that we have, or rule law we have for biology, that DNA codes for RNA and RNA codes for proteins. So we go from DNA to RNA to proteins. So that's why we have DNA in our cells. Comparing DNA molecules and RNA molecules. I want to compare the two. DNA is a double helix. It has a deoxyribose sugar. The nitrogen bases that make up DNA include adenine, thymine, cytosine, and guanine. And the function is that it codes for RNA. RNA is a single strand. It's a ribose sugar. Adenine, uracil, cytosine, guanine. They both have adenine, cytosine, and guanine. RNA will code for specific amino acids. Okay. If you're just compare the two processes of transcription and translation. Ooh, scoot up. Is this going to move? There it is. It's a little slower. All right, where does transcription occur? It occurs in the nucleus of the cell. Who participates in this? We have the DNA. Remember, the DNA opens up, new bases fill in those spots, and it makes RNA. The function is to make an RNA strand. Your outcome, what eventually is released, is going to be a modified strand of mRNA. You had introns cut out, exons left in, you had a cap put on, you had a tail put on. So it is modified before it's released. Translation. Now remember, the amoeba sisters remember them because which comes first, because C comes before L in the alphabet. So try to remember that somehow. So translation happens in the cytoplasm. The participants are the three RNAs. So you have mRNA, rRNA, tRNA, and then you also have the amino acids that have to form a chain. The function of this is to actually make an amino acid chain, which is going to be the polymer. It's going to take monomers to make a polymer to make a protein. So the amino acids are like train cars, and we're connecting all these train cars. The product of this will be an amino acid chain. It is a polypeptide. That's a protein. Right, what role does DNA play in this? It carries the instructions or the pattern for the order of the RNA bases. And then the RNA bases make the mRNA. And it's going to be according to whatever that cell needs to make. You are not going to copy the entire strand. You're going to copy a portion of it. Just a portion will open up to do this. Let's see. I thought I had this drawn somewhere, but I don't. Only a portion of it's going to open. So don't think that each time you make this huge copy of everything. You don't. Okay, so you could have your strand Here's all the rest of the strand, and it's not being read. You're only reading the one little portion you need for that cell. The only time you would worry about when you're making new cells, like if the cell is going through mitosis and needs to copy its DNA for a new cell, then it will copy the entire strand. If it's only copying the DNA to make proteins, it's only going to like copy little portions of it at a time. Nicknames for the DNA strand. I've only heard it called the template strand, but there's also the non-coding strand, minus strand, antisense strand. Walk yourself through the process of transcription. This is easiest if you have the picture with you. Okay, so here I've got the picture. The DNA unzips. The bases line up, the RNA bases line up, they fill up along there. The RNA is released, it's modified, and it exits the nucleus. So I just go in order, like from what happened, you know, it unzips, they fill in, it's modified, it's released. I'm going to have this exact same picture, and I'm going to ask you, when is the mRNA modified? When does tRNA pick up amino acids? When is that protein chain released? Okay. So real basic questions about that. Let's see here. Number 7, walk yourself through translation. So the same kind of thing. The mRNA goes to the rRNA. It attaches and goes through, it actually attaches at the 5' end. It goes through 3 amino acids at a time. Remember, that's called a codon. At the same time, a tRNA picks up the appropriate amino acid, carries it down here, and matches the base, drops off that amino acid in that growing chain. Now the tRNA is released. It can go pick up another amino acid and do the same thing over again. This strand is released, and now it's going to code for that protein, for whatever your cell has to do. It's a protein now. This ribosome can be reused. The bases can't. They can't be reused because now they've attached here. Now the bases on the tRNA, they're still there, and that tells it what amino acid it can pick up. But all of, or a lot of the stuff can just be reused. All right. So the mRNA attaches. tRNA brings the amino acids to the rRNA. The amino acids are dropped off. They make a chain. The chain is released. The amino acid chain, that's your protein. So as that's released, your mRNA strand is also released. It can also be reused. List some of the functions of RNA polymerase. This is really one of the only enzymes. We really only talk about 2 enzymes with transcription and translation, and this is one of them. And we know it's an enzyme because it has an ACE ending, RNA polymerase. It's going to unwind and unzip that original DNA strand wherever you need it opened. And it's going to carry bases to that spot and line those bases up in order to make the mRNA. Ribozymes are also enzymes. These are ribosome particles that act as an enzyme. Their specific job is during modification. They will remove, they actually cut out the introns. So cut out during modification. So they're kind of like scissors. They're going to come in there and they slice it, and then that way the mRNA can come back together. So they're cutting out pieces and pulling it back together, cutting out, putting back together. Compare transcription and translation in prokaryotes and eukaryotes. Well, prokaryotes are organisms that do not have a cell nucleus. So it's going to be bacterial cells. Eukaryotic cells do have a cell nucleus. So transcription and translation have to happen in the cytoplasm of a bacterial cell, but they happen in the nucleus. We have transcription in the nucleus. We have translation in the cytoplasm. So there's two parts in the eukaryote. So the first part and the second part. Remember, there's also rRNA on rough ER. I don't specifically ask you about that, but rough ER is rough because it's covered with ribosomes. So protein can also be made there. Starting points. With prokaryotes, in their string, they have a ring of RNA or DNA. They have one starting point. They're going to have one starting point, and then it goes around there and ends. And one, it just has to start there and end. So if it makes a copy, it has to make a copy of all of its genetic material. In eukaryotes, we have many points on that. When I showed you, I don't know why I drew that. When I showed you, it can open up at any point along the DNA strand. The difference in the actual process, there's a big difference where in bacterial cells, transcription and translation happen at the same time. So we have transcribing the DNA into RNA into protein happens continuously. It all happens at the same time. Ours is in two phases, where transcription happens first in the nucleus and then translation happens in the cytoplasm. So for us, there's two steps. I got a whole bunch of new markers. Oh. So now I need another container to put them in because I have a problem. I'm sorry. I have an addiction. It is office supplies. All right, scooting on up. Describe how new strands of mRNA are modified. You've got the strand, and there's going to be pieces cut out. So we have a strand like this. Do you remember this? And we've got some different portions. Okay, that's what got made in the DNA molecule. That's what we just produced. It's now in the nucleus, it's in the cytoplasm or nucleoplasm, and it needs to be modified. So I'm going to estimate the size of this. It's going to be a little bit longer, I guess. So we're going to have those enzymes come along and the ribozymes cut here, cut here, cut, 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 cut. And when this gets put together, it's going to be red. Pretend this is the same length. I don't know if it is or not. Okay, so those little portions weren't necessary. They got cut out. And then it puts two different caps on here. It puts a cap here, and it puts a tail here. So this is the 5' cap, GTP cap, poly A tail. So 5', and inside that 5' is the GTP. And then the poly A tail. And remember, that's anywhere between like 50 and 200 adenines in a row. I'm trying to think, what do we do next in this class? It's gene expression. Okay. All right, compare the modifications. What are the differences in these things? We have a 5' cap, a GTP cap, and a poly A tail. So the 5' cap gets added to the 5' end of the mRNA molecule. The GTP cap is also added to that end, but it's kind of like inside that first cap. The 5' comes first. And poly A tail gets added at the end. Their functions, the 5' cap works to protect the strand. That strand's going to have to attach to the ribosomal RNA, and it needs to be protected. It's going to be pushed through there, almost like you're lacing your shoes, and you don't want that shoelace to fray, so you have this protector at the end. Same kind of thing when we're looking at the mRNA strand. So it's to protect that strand when it's entering the rRNA. And again, it's at the 5' end of the mRNA. GTP cap, it works to stabilize the strand. It's at the 5' end after that cap, that first cap gets put on. Poly A tail also stabilizes the strand. Now, I promise you on a quiz, I'm not going to say which one of these modifications stabilizes the strand of mRNA, and then give both of those as options. I'm not going to do that. I specifically went through and made sure I don't give you, two possible answers. So I would have one, but not the other. Okay, if it was multiple choice. The poly A tail is at the 3' end after the stop codon. And we're going to learn about codons and how they are 3-letter codes that tell for stopping and starting. Okay, I am recording this, so I'll also put this on Canvas. Oh, I got a headache. Got this, guys? Page 3. Comparing introns and exons. Oh, what's the difference? What's the purpose of each? Okay, an intron is genetic information that's not used in a specific protein. That doesn't mean it's garbage. It used to be, people used to think it was junk DNA and it just was there. We ought to know better. Most of the things that we have are going to have a purpose. So what can happen is these little portions could be on-off switches. They could be information that could come into play, and maybe we don't know about them yet. Maybe we're not sophisticated enough, or our technology's not sophisticated enough to know what it does. But there is a purpose to this extra information. What happens to it? It gets cut out when that mRNA strand is being modified. Exon, it is the genetic information that's used to actually make that specific protein. Now, think about it. There could be a time when the RNA is modified to take something out. The next time it could be modified to leave that information in. There could be possibilities where that same strand of DNA could actually code for different things. And that adds a lot of variety to a species. All right, why do biologists believe that all living things share a common ancestor? This is a question we have in almost every unit because it's evolution and common ancestry is a huge theme. It's like the top theme in AP Bio. So what are some of the things that we use as evidence for common ancestry? That all living things have DNA and or RNA. We share the same base pairs, adenine, thymine, cytosine, and guanine in DNA, adenine, uracil, cytosine, and guanine in RNA. We are made of the same macromolecules, carbs, lipids, proteins, and nucleic acids. We have the same nucleotides. Okay, all of our nucleotides are that phosphate group, sugar, nitrogen base. They're just in different orders. Last hour, I don't think it was you guys, last hour we were talking about how much DNA we have in common with other organisms. We have 99.9% in common with other humans. We have, just a second, I think it's 96% with bonobos monkeys and chimps. 50% of our DNA is identical to bananas.
That's concerning.
80% with cows, 61% with fruit flies. Let me see how much potatoes. What? We have 80% of the genes found in potatoes?
Well, actually, maybe that kind of makes sense.
Potatoes? I'm in shock.
Because potatoes have a lot of machinery to make a ton of different like things that humans need. They're one of the most nutritious foods, so maybe they have a lot of the genes needed for processing and handling those things, similar to humans. I don't know.
That's pretty amazing.
You are 80%.
The lowest amount. Yeah, we only share 18% with yeast. And then other little protozoans and stuff we share just very small amounts. But we are all made out of the same bases. They're just in different orders. So that's pretty cool. I don't know. All right. So what are retroviruses? This is, I don't show you a picture of one, but I do ask questions about a retrovirus. And that's because the AP, they think you need to know this.
HIV is a retrovirus, right?
Yes. All right. That's what the retrovirus would look like if we're looking at COVID. These are little attachment points where it attaches to living cells. This is a protein coat. It's got just like a little bit of genetic material. It cannot grow on its own. It cannot reproduce on its own. It does not eat food, none of that. What has to happen is this has to attach to some kind of body cell or living cell. It injects its DNA into there, hijacks the cell. The cell becomes like a zombie and does whatever that DNA tells it to do. That's what's going on in your body when you get any kind of virus. They inject their genetic material. And since they're called retroviruses, that tells us it's RNA. That living cell is now kind of hijacked. It's now kind of a zombie doing anything that viral RNA tells it to do. And it's going to make more viruses. So now your cells actually make more viral cells. Sometimes this is harmful. So sometimes it could be a harmful issue where the virus could make you very, very sick. It could be cause death. There are other viruses you don't even respond. Your immune system just like takes care of it. One type of virus, RSV, is respiratory syncytial virus. When little kids get it, like babies and toddlers get it, they can die. They get horrible respiratory infections. we are come in contact with it all the time. And so we've already built up an immunity. Typically, we've had it when we were little. Maybe we had a real mild case and we built up immunity. So we all should be carrying the immunity to RSV. These, what?
Actually, I thought retroviruses use like DNA. They use their DNA and they implanted it into the genome of the cell that they hijacked.
All viruses do that. All viruses inject their genetic material. Let me look this up for sure. They're all going to do, that's the only way they can reproduce, because viruses are not alive.
Well, I mean, I thought what some did was they used RNA and they essentially got it so that the cell machinery kept copying their RNA instead of looking at the DNA of the cell itself.
Okay, this is confusing. There are a group of RNA viruses. They insert DNA into the genome. So that really makes no sense. Why are they called an RNA? It uses RNA as its genetic material. When a retrovirus infects a cell, it makes a DNA copy of its genome that is inserted. Okay, so it has RNA as its information. It makes a DNA copy, injects it into the new cell. Well, that's kind of a beast.
What?
Wow, it's kind of mean, you know. I'm going to tell you some of them that are retroviruses. Just a second. And there's all kinds of other viruses too. Oh, let me think. There are some leukemia viruses, HIV. There's all kinds of animal diseases. Ugh. All kinds of animal viruses. That's really gross. Feline leukemia, equine foamy virus. I don't know what that is. It's something in horses. Gross. That's really gross. All right. So wait, no, one more thing. Page 4. I'm showing you this picture. This is the exact picture on your quiz. All right, so these are some questions. When does the tRNA molecule pick up the correct amino acid? 1, 2, or 3? It happens in this middle part. I'm going to say 2. RNA bases move to the appropriate spots on the DNA template strand. When do I see that DNA strand? Over here. 1. mRNA detaches from rRNA and can be reused. When do I see both the mRNA and the rRNA? Right here. This is mRNA. This was our RNA. This can be reused. Number 3. The 5' cap of an mRNA strand attaches to the rRNA. It has to be here. Wait, I put down the wrong number. The 5' cap attaches to the rRNA molecule. That would be 2. I read it too fast. The mRNA strand is modified. Back here, besides the fact that I have the word modification. 1. All right, so I'll give you this drawing and ask you really basic questions about the drawing. Any questions?